Morning. Today we will study about fate of pyruvate or pyruvic acid. Different pathways for cellular respiration. In this pyruvic acid, there are two different ways to make energy in cell. In determining the fate of pyruvic acid after glycolysis, two questions arises in our mind. First, is there oxygen present or absent? For example, aerobic or anaerobic conditions. And second, are there any mitochondria present to facilitate TCA or tricarboxylic acid cycle, and ETC means electron transport system chain. Fate of pyruvate in aerobic conditions. In aerobic conditions, with mitochondria present, the two molecules of pyruvate produced from the molecule of glucose is then transported to the mitochondria of the cell where it enters the TCA cycle. One molecule of glucose converts then to pyruvate via the TCA and ETC electron transport chain. Energy is then generated and NAD plus is generated in the ETC electron transport chain. In animals, plants, and many microbial cells having this system for aerobic conditions. This is the overall structure of aerobic conditions. Fate of pyruvate. Pyruvate enters in mitochondria, gone through the TCA cycle, then electron transport chain, and produce energy. Energetics of aerobic conditions. In aerobic conditions, two pyruvate molecules are obtained from one molecule through glycolysis. Each of the pyruvate yields one NADH. Thus, there are two NADH molecules to be oxidized through the electron. Of NADH yields 3 ATP molecules. Thus, a total of 6 ATP molecules are produced at the level of PDH complex or pyruvate dehydrogenase complex transport chain. Means the energy produced in aerobic conditions. On the other hand, the fate of pyruvate in anaerobic conditions, there are two pathways. Glucose, which converts into pyruvates by glycolysis, either converts in ethanol or in lactate or lactic acid. In ethanol, it converts in two molecule of ethanols and two molecule of carbon dioxide, and fermentation to ethanol in yeast is the best example of hypoxic or anaerobic conditions in yeast. Anaerobic conditions in higher animals, fermentation to lactate in vigorously contracting muscles, in erythrocytes and in other cells, and in some microorganisms also. First, ethanol fermentation. In this process, under anaerobic conditions, pyruvate is further metabolized to ethanol with the regeneration of NAD plus and formation of carbon dioxide. Here we can see the carbon dioxide released and the NADH consumed, and final product is ethanol. One molecule pyruvate, one molecule ethanol. In a simple eukaryotic cells like yeast, pyruvate is converted to ethanol with a liberation of carbon dioxide in two-step fermentation process. These steps are irreversible reactions where catalyzed by pyruvate decarboxylase and alcohol dehydrogenase. TPP, thiamine pyrophosphate, is a cofactor for both these enzymes. In a first step is catalyzed pyruvate decarboxylase, in which pyruvate is decarboxylated and converted to acetaldehyde. The product acetaldehyde formed in this reaction serves as the substrate for the next enzyme in the pathway. Pyruvate decarboxylase requires thiamine pyrophosphate or TPP and Mg plus 2 as coenzyme and a cofactor respectively. Thiamine or vitamin B1 contained in the thiazolium ring and serve as a source of thiamine pyrophosphate. In second or other anaerobic conditions, pyruvate converts in lactate. The reduction of pyruvate by NADH to form lactate is catalyzed by lactate dehydrogenase. The reaction takes place in the cell where the amount of oxygen is limited, as in muscles during expansion. Here we also can see the NADH is consumed during this process. So the energetics of anaerobic conditions, two pyruvate molecules are obtained from one glucose molecule through a glycolysis. Each of the pyruvate uses one NADH. Thus, there are two NADH molecules used during the anaerobic conditions. Each of NADH gives 3 ATP molecules. Thus, a total of 6 ATP molecules are used at this level. Energy used in anaerobic conditions. Effect of pyruvate in aerobic and anaerobic conditions. In a nutshell, we can see here anaerobic conditions, NADH consumed. In aerobic conditions, NADH produced, and this alcoholic fermentation and aerobic conditions, NADH used. The lactate and ethanol produced by microbial fermentations are commercially exploited by the human use. Thanks.